now. Now, our next speaker, Dr. Supriya Bhattacharya, is a actually senior student, and he will be speaking his work for us, for the students also. So in a way, we are fulfilling the core purpose of the workshop grant. Supriya Bhattacharya is a senior PhD student at the Israeli Faculty of Medicine, Bar-Ilan University, located in Sfat, in the north of Galilee, in Israel. He has obtained his master's degree from this institute, from biochemistry division. His master's thesis was development of a diagnostic test for the detection of classical swine fever virus. He continues to remain on the virology, virus research. For his PhD work, he is currently working under Professor Meir Shammai, and today he will be sharing his work and giving a talk. Title is CRISPR dCas9 assay for identifying the directionality of protein recruitment in cells. Ladies and gentlemen, please welcome Dr. Supriya Bhattacharya.
Thank you, sir.
Stage is yours.
So I will start sharing my screen.
Yeah.
Can you see my screen now?
Not yet.
Okay. Now you can see?
Yeah, you can. We can.
Okay. So thank you, everyone. And I want to thank you, the organizers of the stem cell workshop, so to give me an opportunity to present my PhD work, part of a PhD work that I did during the last 4 years. So this project, which is we call CRISPR dCas9 based assay for identification of directionality of protein recruitment in cells. This actually I did with another PhD student in our lab, Idolavi, and I will be discussing about this project today. So in our lab, mainly we work on two gamma herpes viruses that causes malignancy in humans. One is Kaposi sarcoma associated herpes virus, and one is a Epstein-Barr virus. Now, both the viruses belong to gamma herpes virus family, and especially the Kaposi sarcoma associated herpes virus is a double-stranded DNA virus, which is 165 kilobase pair in length, and it actually present as a circular viral episomes in the cell. It actually present as a circular viral episome in the cell. And this virus has two phases. One is latency and one is lytic phase. Now in latency, very few gene expressed in the cell, very few gene of virus actually express, and various type of stimuli like hypoxia or environmental stress, when it will affect the infected cells, the virus will go to lytic cycle, and you can see here, and it start producing the infectious variants. You can see it here. Now, this is the list of all the pathologies or the cancers that it causes in human. And in my work today, I will mainly focus on KSHV, Kaposi sarcoma. And in our lab, especially we work on latency, not the lytic phase. The reason behind it, because when the KSHV causes the cancer, most of the cells remains in latency. So we wanted to check how the latency actually helps to develop cancer in case of KSHV. So this is all the list of diseases you can see here that are caused by KSHV and EVV. Now, before going in more detail about my presentation, I want to remind everybody or refresh their memory about CRISPR. I know everybody has an idea about CRISPR technology, but to give a brief introduction about CRISPR, if we can imagine that one of the most important application of CRISPR in today's mammalian cell culture is its use in insertions of a new genomic sequence in a mammalian genome, or it's wisely used for knockout of a particular protein coding gene. Now, you can see here is the Cas9, and here is the S-guide, and S-guide has a 20 base pair specificity determinant, which actually determines where you are targeting the S-guide. So you can actually target anywhere in the human genome just by changing the 20 base pair here, and rest of the sequence of the S-guide remains same, which actually also known as the adapter sequence for S-guide. Now, when we transfect or transduce the Cas9 and S-guide together in a cell, it will go to the region where you want to direct, and it will create a double-stranded break. As you know, that Cas9 is a nuclease, so it will create a double-stranded break. And after creating the break, there is two possibilities. One possibility is if you provide a donor DNA, so there will be a DNA recombination and there will be an insertion of a new sequence in the genome. But if you don't provide a donor DNA, cell will try to repair that DNA damage. And the way it repairs, it's called non-homologous end joining. So when it happens, non-homologous end joining, this is very error prone. And when the non-homologous end joining occurs, it actually creates insertions or deletions. Now, if you know that every protein coding gene has a particular open reading frame. So now if there will be insertion and deletion, there will be a frame shift. And this frame shift will create a premature stop codon. And when there will be a premature stop codon, there will be no protein anymore. In that way, we can use CRISPR to knock out a particular gene. Now, another very important example of CRISPR nowadays is CRISPR imaging. In that case, we don't use Cas9. Instead, we use a dead Cas9, which is unable to cut DNA. But with the S-guide sequence, we can direct it actually anywhere we want. So here you can see an example where they actually fluorescently tagged one or two base pair of dCas or S-guide, and they marked the telomere. And you can see effectively that how beautifully it marked the telomeres in a live cell. And also you can tag the S-guide, and also you can tag the dCas, and by that way you can actually mark a particular genomic loci in cell. Here in the example, you can see they mark telomere, and MAS means major satellites. So you can see how beautifully we can mark or image a particular region of a genome using CRISPR technology. So in my PhD study, I used CRISPR imaging technique, but I went another step forward, and I used the CRISPR technology to determine actually the directionality of protein recruitment in cell. So to explain that a bit more, I will just give you a simple example. Let's say there are two proteins, protein A and protein B, that are present in cell. And by co-immunoprecipitation assay, you can determine that yes, protein A is interacting with protein B, but protein B also interacting with protein A. But previously there was actually no method to determine which one is a dominant partner between protein A and protein B. With our newly developed assay, you can actually determine which is a dominant partner in a protein-protein interaction. So in today's talk, I will divide my talk in two parts. Part one is the establishment and validation of CRISPR-PITA. PITA is protein interaction and telomere recruitment assay. We named it like that because PITA actually is a name of a bread here in Israel. So to make it a bit funnier, we named it PITA. PITA means protein interaction and telomere recruitment assay. And this methodology can be used to determine the directional recruitment between proteins. And in the part two of my talk, I will tell about how I used this newly developed method to answer a very critical question in KSHV biology. So let's go deep into part one. So before going into deeper, I want to tell something about KSHV and its latency. So you can see here, the KSHV actually a double-stranded circular viral genome, and it actually present as a episome in the cell. It doesn't integrate into the genome. Now, to be present in the infected cell, it must tether with the host cell chromosome. Otherwise, during the time of cell division, if it doesn't tether with the host cell chromosome, it will be flow away from the cell, and the genome will be eventually lost from the infected cell. So in order to tether with the host cell chromosome, virus produces LANA. LANA is a latency-associated nuclear antigen, which actually binds with the viral genome, and then it connects the viral genome with the host cell chromosome. And by that way, LANA helps the virus to maintain the genome in KSHV infected cells. Now, as many molecules of LANA actually binds, you can see here, there are many molecules of LANA that binds with the viral genome. So it actually creates a dot in KSHV infected cell. So you can see here, for comparison, I put a figure where you can see this is a KSHV infected cell, where you see very beautiful LANA dots, and each dot represents a KSHV viral genome, viral episome. And when there is now this is the infected cell, but when we take LANA and infect and we transfect it in a cell, in a non-infected, non-KSHV infected cells, you can see it creates a diffuser pattern. It doesn't create those dots. So so this actually make it very difficult to do a protein screen, because if you want to screen the proteins that actually LANA interacts with, you actually cannot do it in a non-infected KSHV non-infected cells, because there is no LANA dots. Because in non-infected cells, KSHV, this actually present as a diffuse LANA. So in order to create LANA dots in KSHV in non-infected cells, we use this method we named as CRISPR-PITA. Now here what we did, here you can see this is a dCas. Now this dCas is actually fused with a SUNTAG. Now this SUNTAG has 10 different epitopes. Now you can, in our case here we took LANA, but you can take any protein you want and you can fuse it with a FC. Now this FC region will actually bind with those SUNTAGs. So in that way, actually you can gather up 10 molecules of LANA in a particular place, because this actually contains 10 repeats. And so there will be 10 molecules of LANA that will bind here. Now this whole complex, you can guide to any particular region in the genome with S-guide. In our case, we directed it, so you can direct it in a unique sequence, like let's say in a particular promoter, or you can direct it to repeat sequence like telomere or centromere. So in our case here, we directed it to the telomere. Now in panel B, you can see that when there is no LANA, we don't see any LANA. But when we put LANA without the SUNTAG, just we put the S-guide and LANA FC, and you see that there is diffusive LANA. But when we put SUNTAG, you can see very beautiful LANA dots that are formed in telomere. So in that way, we successfully actually created LANA dots in KSHV non-infected cells. Now, after successfully creating those dots, we performed the assay called PITA, in which we ask the directionality of protein recruitment. Now, this actually represent the telomeric LANA dots. Now, let's say you want to see if any LANA has the ability to recruit any protein, any endogenous protein in the cell. So if there will be a recruitment, you will see that the distribution pattern of that endogenous protein will change and it will co-localize with LANA. But if there will be, if LANA will be unable to recruit that protein, you will see there is no change in distribution pattern of those cell of endogenous protein. So now let's discuss about some proteins that we used in this assay. So in the literature, when we searched the literature, we saw that there are 3 proteins, ORC2, SYN3A, and MSCP2. These 3 proteins actually interacts with LANA. This is already reported in the literature. So we wanted to check if now these 3 proteins are actually can be recruited by LANA in our assay. So you can see here in the left side of the figure, this is the different component we transfect in the cell. And in the right side of the figure, you see a histogram or a fluorescent intensity map that represents the intensity of different fluorophore across the white line. And here throughout the assay, the protein that we actually direct to the telomere will be in red, and the endogenous protein, and the protein we ask if it's recruited by LANA or not, that will be in green. So here you can see when we don't put dCas9 tag, there is a distribution of LANA throughout the nucleus, no dots. And you don't see any change in distribution pattern of those cells, of the endogenous ORC2. But when we created LANA dots with the sun tag and we send it to the telomere, you can see there is change in distribution pattern of ORC2. And actually, it co-localizes with LANA. You can see from the peaks in the histogram. So from this experiment, we concluded that actually LANA has the ability to recruit ORC2. The same assay we did with SYN3A, and you can see when there is no sun tag, LANA is distributed throughout the nucleus. But when we created LANA dots inside the in a telomere, and we see that LANA is actually able to recruit SYN3A, and you can also see the histogram of colocalization between SYN3A and LANA. Then we asked MECP2. So MECP2 is also a known interacting, from the literature we know LANA and MECP2 interact. So in our assay, we wanted to check if now LANA have the ability to recruit MECP2 to the telomere. So here you can see that we created when there is no sun tag, LANA is distributed throughout the nucleus, and there is no change in distribution pattern of the endogenous MECP2. But when we created LANA dots, still there is no change in distribution pattern of MECP2. That means LANA is unable to recruit MECP2 to the telomere. So although from the literature we know that LANA and MECP2 interacts, but in our case, we couldn't manage to see the ability of LANA to recruit MECP2. So here we did a Pearson correlation coefficient of all the 3 proteins that we mentioned, ORC2, SYN3A, and MECP2. Now, as the protein that we are using here are known LANA interactors. So when there will be no sun tag, what we expect is they will interact with each other. There will be high Pearson correlation coefficient because they are already there. We know that they are interacting with LANA from the literature. But when we direct LANA to the telomere, so when the LANA will have the ability to recruit that protein to the telomere, then only we will see an increase in Pearson correlation coefficient. And if it fails to recruit, then we will see actually a reduction in Pearson correlation coefficient. And that's what we exactly see here. You can see with ORC2, when there is no LANA, actually, when there is no sun tag, there is high correlation coefficient. But when we put sun tag, there is increase in correlation coefficient because LANA has the ability to recruit ORC2. In SYN3A, Also, we see that it's almost similar, like with Suntag and without Suntag. But in case of MECP2, as LANA actually fails to recruit MECP2, we see actually a reduction in Pearson correlation coefficient to when we direct LANA to the telomere. So from all these experiments, we can conclude that LANA is able to recruit ORC2 and SYN3A, but LANA is unable to recruit MECP2. So one question that that came into our mind is, we know what is the function of MECP2. Now, MECP2 is actually a methylated DNA binding protein. It binds with the methylated DNA in the gene in the cell. So, so one possibility that actually arises in our mind is as the affinity between MECP2 and methylated DNA is very high, so LANA is actually unable to break that interaction and can recruit MECP2 to the telomere. So in that case, what we did, we created a HCT double knockout cells where we knocked out DNMT1 and DNMT3A, and these cells actually doesn't contain any methylation in the genome. So if there is no methylation, so MECP2 will be free. Now we wanted to see if now LANA can recruit that free MECP2 that is there in the cell, in those cells. So you can see even in HCT DKO knock cells, telomeric LANA fails to recruit any MECP2. And this is actually the wild type of HCT. We showed it as a control that in the HCT wild type also, there is no recruitment of MECP2 by LANA. And also when there is a double knockout, then also LANA fails to recruit MECP2. So then we actually wanted to confirm our model. We wanted to see that whatever we are looking at is actually true. So for that, what we did is, we took KSHP negative BJAB, which is a B-cell lymphoma, and KSHP positive BCBL1 cells. And we stained for the proteins that we used in our CRISPR-Pita assay. And you can see here, we can see very nice colocalization of ORC2 and SYN3A. But we don't see any colocalization between LANA and MECP2. So this result actually confirms that whatever we actually see in KSHV infected cell, that we also see in our CRISPR-Pita assay. Now, what can be the reason that two protein that we know from the literature that interacts with each other, LANA and MECP2, but in our Pita assay, we don't see that LANA is able to recruit MECP2. So from that point of view, we thought maybe it's a matter of directional recruitment. Maybe only MECP2 has the capability to recruit LANA, not vice versa. So to test that, what we did now, we instead of LANA FC, now we created MECP2 FC, and we directed MECP2 to the telomere, and then we transfected GFP LANA, and then we see that when the MECP2 is directed to the telomere, it actually has the ability to recruit LANA. So you can see from the histogram also that that there is colocalization between MECP2 and LANA. So this is without Suntag. So there is equal distribution of MECP2 throughout the nucleus. And when there is no MECP2, there is no change in distribution pattern of LANA. And when only you put MECP2, it actually changes the distribution pattern of LANA, and it can recruit LANA to the telomeres. And here, here we used GFP as a negative control to see that actually LANA is going there, not the GFP. It's actually used as a negative control, and you don't see any change in distribution pattern of GFP. then we wanted to verify which region of LANA actually interacts with MCP2. So here we did the same thing. We actually directed MECP2 to the telomere, and then we transfected LANA N plus C terminus, LANA C terminus, and only LANA N terminus. And you can see that LANA N plus C and LANA C actually can be recruited by MECP2 successfully to the telomeres. But on the other hand, LANA N terminus actually doesn't change its distribution pattern and doesn't go to MECP2. So from that, we concluded that here actually you see the statistics for Pearson correlation coefficient, and you can see LANA N has much less Pearson correlation coefficient than LANA N plus C and C. So from that, we concluded that actually MECP2 can recruit LANA via its C terminus. Now, the people who actually work with him, the figure still I showed you is all immunofluorescence. So now, if you work with immunofluorescence, you already know, I guess, that there is many false positive results. And here we are directing something to the telomere, and we are transfecting huge amount of protein in the cell. So anyway, it will come into your mind if it's true in a single promoter level. So to verify that, we did a chip assay, which is also called a chromatin immunoprecipitation assay. And for that, what we did, actually, we chose ERBB2 promoter, which is also known as HER2 promoter, which is very popular in breast cancer. It amplifies and all. So we took the ERBB2 promoter and we directed LANA via Suntag to this promoter. And here 1, 2, 3, 4 are different treatments. First treatment is we direct LANA to the telomere, to ERBB2. And the second, there is a blank vector. In the third, we direct MECP2 to that ERBB2 promoter. And the fourth treatment, and we also transfect LANA. And in the fourth treatment, we only put LANA. So in third treatment, we only put actually LANA, and in fourth treatment, we direct MECP2 to the promoter. So this is the LANA chip, and this is the MECP2 chip. And you can see when we direct LANA to ERBB2 promoter, you can see significant enrichment of LANA there in that promoter. But on the other hand, there is no much enrichment of MECP2. And treatment 2 and 3 are only blank vector and LANA. And in treatment 4, you can see when we directed MECP2 to that promoter and we transfect LANA, actually LANA has MECP2 has the ability to bring LANA, and you can see high enrichment of LANA over here. So the results that we actually showed by immunofluorescence assay is actually also true in a single promoter on a single locus, that the unidirectional recruitment. So from all these experiments, what we can conclude, the interaction between LANA and MECP2 is unidirectional, and MECP2 is the dominant partner here. MECP2 has only the ability to recruit LANA, and on the other hand, LANA is unable to recruit MECP2. So still now we talked about a viral protein and a cellular protein interaction. So what will happen in case of two cellular proteins? Is the directional recruitment will work in case of two cellular proteins also? So one of the very good interaction that we know that happens in a cell is MECP2 and HDAC1. Now, MECP2 is actually a repressor. It actually binds with the methylated DNA and it represses the expression of a particular gene. And HDAC1 is actually a co-repressor. So what we expect here, a co-repressor is actually used by the other repressor. So we actually will not expect that HDAC1, which is acting as a co-repressor, it needs to act with other repressors. So if HDAC1 will have the ability to recruit MECP2, so wherever HDAC1 will go in the genome, it will bring MECP2 with it, which is not possible. On the other hand, MECP2 will acting as a repressor and will have the ability to recruit co-repressor HDAC1. So here in this case, we expect actually that MECP2 will recruit HDAC1, but HDAC1 will be unable to recruit MECP2. So we again did the CRISPR-Pita assay. We sent MECP2 to the telomere and we stained for the endogenous HDAC1. And you can see from the from the histogram of the intensity of the fluorescence that they are colocalizing. Actually, MECP2 able to recruit HDAC1. But on the other hand, when we sent HDAC1 to the telomere and we wanted to see if now it can recruit MECP2, actually you don't see. And there is no recruitment of HDAC1 by MECP2. So this is the statistics of this experiment, and you can see there is a significant reduction in Pearson coefficient when we measure the ability of HDAC1 to recruit MECP2. But on the other hand, you can see that there is an increase in Pearson correlation coefficient when we direct MECP2 to the telomere and measure the HDAC1 recruitment. So this is a graphical representation that when MECP2 bind with the DNA and recruit HDAC1, and by that way it can repress the expression of a particular gene. So from this experiment, what we can conclude is that MECP2 able to recruit HDAC1, but HDAC1 is unable to recruit MECP2. Then how we can actually explain this directional recruitment? How we can explain this recruitment in a cell? How we can explain this directionality? So one possible explanation can be the availability of domains during the time of interaction between those proteins. So if we look at MECP2 structure, MECP2 has an N-terminal domain, has a methyl binding domain, has a transcription repression domain, and also a C-terminal domain. Now, N-terminal MBD, and now MECP2, if you look here in the figure bottom, MECP2 can exist in two forms in the cell. One is free form in the solution, which exists as a monomer, and in that case, the MBD and TRD domain are not available. But when it's bind with the DNA and then it dimerize, and so you can see its DNA and its binding, then these domains are available, and then it can actually interact with the protein. So if you look here, both HDAC1 and LANA, they interact with MBD and TRD domain of MECP2, and which are not available when MECP2 is in free form. So maybe that's why we didn't manage to get any colocalization between LANA and HDAC1. But on the other hand, there is another protein in the cell, which is called HP1 alpha, which is heterochromatin protein alpha, which binds actually with the heterochromatin. It actually interacts with the N-terminal domain of LANA, which is actually available when MECP2 is present in free form. So we hypothesize that maybe HP1 alpha will have the ability to recruit MECP2. So what we did, we again performed the CRISPR-Pita assay. We directed HP1 alpha to the telomere, and then we checked now if HP1 alpha can recruit MECP2. And exactly that's what we found. You can see from the histogram here and also from the statistics that HP1 alpha actually can recruit MECP2. So from all this experiment, what we can conclude is that when MECP2 is in free form, there is no MBD TRD domain available, and that's why LANA and HDAC1 is unable to interact with MECP2. But when MECP2 actually binds with the DNA and the MBD and TRD domain will be available, then LANA and HDAC1 will be able to interact with MECP2. And here you can see the statistics, and you can see there is an increase with the SUNTAG when you direct it to the telomere. So if that is true, that LANA actually can interact with MECP2 only in its DNA-bound form, so we decided to verify that. We decided to check our hypothesis. So you can see here, there is MECP2 in free form, which is MBD and TRD domain are hidden. Now you can see in DNA-bound form, MBD and TRD domain are available. So we hypothesize if MECP2 now DNA-bound, then it will open the by interacting domain between MBD and TRD, and then LANA can interact. So now if we go back in our figure here, you can see that MECP2 actually can recruit LANA in our CRISPR-Pita assay. So how is it possible? So if that is true that only LANA can, MCP2 can interact with LANA in DNA-bound form, but how we are seeing the recruitment of LANA by MCP2 in our Pita assay? So what we hypothesized, maybe that because SUNTAG has 10 epitopes, okay? So although in our CRISPR-Pita assay, we does not directly measure the binding of DNA by MCP2, but what we can hypothesize is that as the SUNTAG has 10 epitopes, and we are gathering 10 molecules of MCP2 in a very small place, maybe it actually mimics a dimerization. So to test that, what we did, actually, we only took the SUNTAG part and only took FCMCP2, and we transfected it in a KSHV infected cell. So here you can see this is a KSHV infected cell, and this is the LANA dots in KSHV infected cell. Now, when we only put SUNTAG, we don't see MCP2. But when we put only MCP2, you can see it's equally distributed throughout the nucleus. But when we put SUNTAG and MCP2 together, then maybe it actually mimics the dimerization and opens the binding domain of ambient TRD through which LANA can interact. So you can see here, when we put this together, it actually forms dots and goes to LANA dots. So from this, actually, we can conclude that with the help of DNA, we can actually break this unidirectional recruitment. So here you can see MCP2 is not associated with LANA dots in infected cell when there is no SUNTAG, but when we put SUNTAG, it actually forces the dimerization in SUNTAG, and that leads to recruitment of MCP2 to the LANA dots. So from all this experiment, we can conclude that MCP2 only can interact with LANA in DNA-bound form. Now, after telling you all this, I want to now use this technique to answer a very important question in KSHV field. Now, what can be that question? If you remember my beginning slides, I told you that KSHV is a viral genome. It is circular, and it actually present as an episome in KSHV infected cells. Now, in order to maintain the genome in KSHV infected cell, it must tether with the host cell chromosome. Now, LANA actually binds with the viral genome, but LANA also needs an anchor in the host cell chromosome so that it can tether with the host cell chromosome. Now, what we found is actually that LANA binds with the episomes, actually LANA binds with the MCP2 only in DNA-bound form, but not the free form of MCP2. So this actually makes MCP2 is a perfect candidate for an anchor. So LANA maybe will bind with the DNA-bound MCP2, and by that way, it actually helps the virus to tether with the host cell chromosome so that it will not be lost during the cell division. Now, to test that, what we did, we actually took a cell line from Rett syndrome patients. So what is Rett syndrome? Now, Rett syndrome is a neurological disorder that happens in childhood, and it actually happens because of different mutations in different region of MCP2. Now, what we did, we took 3 different Rett syndrome cell line. One cell line that expresses wild-type MCP2, one cell line that expresses this mutation T158M, and another cell line that expresses another mutation 803 del G, which actually reside outside the MBD and TRD domain. And then we took, and this is T158M mutation. This mutation is very important because this mutation actually prevents MCP2 to bind with the methylated DNA. So we take all these 3 cell lines, wild type MCP2, T158M del G, and 803 del G, and we infected them with KSHV. Now, here we used a recombinant KSHV virus, which actually expresses GFP when we establish latency in a KSHV infected cell. So after infecting all these 3 cell line with KSHV, we actually measure the percentage of GFP positive cell in indicated time points. And you can see wild type and 803 del G successfully maintains the KSHV genome, but T158M, which actually unable to bind with the methylated DNA, fails to act as an anchor for LANA with the hostel chromosome, and it loses the genome very quickly. Here also we measure the KSHV copy number by qPCR or real-time PCR, you can say. And you can see wild type MCP2 and 803-delG successfully maintains KSHV genome, but when there is T158M mutation, it fails to maintain the genome. Now, to support that, what we got in LCLs, we actually used another cell line called NIH33. So NIH33 is a mouse cell line, and one of the unique feature of the cell line is you can see from the structure of nucleus, it has heterochromatin dots. And another important feature of this cell line is doesn't express any MECP2. So you can see when we transfect MECP2 on those cells, it goes to those heterochromatin dots and creates a dot without any pita. So without any pita assay, we can successfully create the dots in those cells, in those cells. Now, we used 3 different mutations of MECP2. One is wild type MECP2, one is T158M, another is MBD deleted domain. So you can see when we transfect wild type MECP2, it can actually recruit, and we also put GFP LANA in all these cases, and it actually can recruit LANA to the heterochromatin dots. But when we put T158M, and also when we put del MBD, we can see that it doesn't bind to the heterochromatin dots, and in turn, it also fails to recruit LANA to the heterochromatin dots. So from that, all these figures, and here is the same Pearson correlation coefficient we did for all these 5 treatments. And you can see in case of wild type, there is high correlation. And here actually we correlated not the green and the red, but the DAPI dots with the GFP dots. Because if MECP2 will recruit LANA, we will see that it will go to the DAPI dots. So here we correlated between blue and green. And you can see in case of wild type, there is very high correlation. In other cases, we don't see any correlation between them. So from all these experiments, we concluded that when there is deletion of methyl binding domain on when there is T158 mutation, it actually doesn't support KSHV latency, and it loses the genome very quickly. Then we did another experiment. Here what we did, we took actually already KSHV infected cell. First, we take, this is ISLK, which is called inducible SLK, which is a cell line that we use routinely for KSHV research. And what we did here, we knocked down first MECP2, and you can see the validation of MECP2. And then after knock it down, we put for colony formation assay to see if knockdown of MECP2 actually affect this cell line. And you can see that when we knock down MECP2, it doesn't affect the growth of this cell line, and it actually form equal number of colonies from the statin colony count, you can see. So we took this cell line, and then we infected them with KSHV, and you can see here, and we knocked down MECP2 in those cells. Now, we infected these cells here with a virus which is called KSHV BAC16, which has a hygromycin resistance. So if our hypothesis is correct that knockdown of MECP2 will actually reduce the genome copy number in cells, so what we will see is that when we knock down MECP2 and we put the selection reagent hygromycin, it actually kills the cell, and we will see less colonies. So we'll took those cells, we already, again, we validated the knockdown, and then we put it for a colony assay with high amount of hygromycin. And you can see only control has colonies, MECP2-1 and MECP2-3 has very lost colonies. And it has also, we quantified the colony number. So from all these experiments, we can conclude that knockdown of MECP2 actually inhibits the latency maintenance. And in KSHV loses genome very quickly when there is no MECP2. So this is our model from all this research. Actually, there is a research from Skinske very long before, in 2006. He showed that LANA need to tether the LANA N terminus of LANA actually need to tether with the histone H2A and H2B. And if you remember our slide from this figure, you can see that N terminus actually doesn't interact with MECP2. So actually what we think actually the C terminus of LANA actually interacts with MECP2. So actually virus uses both histone H2A and H2B through its N terminus, and MECP2 through its C terminus to tether tightly with the hostel chromosome so that it will not be lost during the cell division. So from this, all these results, what we can conclude that we can describe a broadly applicable protein recruitment assay based on CRISPR dCas9, which we termed as CRISPR pita. And recruitment does not always work bidirectionally. In many cases, it is actually functionally unidirectional. And with this assay, we actually identify MECP2 as a critical anchor for KSHB genome, and which would not be identified by any other method. So here, this is a drawing by my professor's daughter. She is 8 years old, and she explained it, I think, very nicely, that this is a DNA bound with MECP2, and LANA is coming with the viral episome, and they, I think, hug with each other to stay in infected cells. Thank you. And this is all the lab members we have. And I thank Idolavi and our Professor Meir Samoy, all the collaborators. And this is actually our view from the lab window. This is the Sea of Galilee. Thank you. I am open to any questions if you have.
Thank you, Dr. Bhattacharya. Supriya, thank you very much. So now, I will switch to the participants to check if they have any queries or clarification they need. Anybody amongst the participants? If not, I would start with this one single question.
Yes, sir.
So, is that the whole recruitment process, is it linked to only the latency, or is it a generalized procedure that beyond the viral interaction, whether we can also use this technology or technique to understand any other cellular phenomena, cell biology in which two proteins are recruited one after another? Is it
Yes,
a general?
Yes, of course. I already showed in a slide about two cellular proteins, MECP2 and HDAC1. And you can use actually this technique in any cell, in any, what you just need to, I will explain it in a bit better way. What I actually did, there is a plasmid, and in this plasmid there is Fc, and you can clone any gene you want. And then you can put the three plasmids together in a cell, and you can check the directional recruitment. So if you are asking that, yes, if you can use it for any cells, yes. And also I showed in these experiments, MECP2 and HDAC1. These two are two cellular proteins. And these two proteins, and this assay actually works on those two proteins also. Okay, now to put it in a bit context, maybe people are still confused about the directionality and all this. So I will just give you a simple example. I hope everybody knows about transcription, right? RNA pol2 eukaryotic transcription. So if you remember the steps in the transcription, first what happens? First, there will be a topoisomerase or gyrase, they will open. Then there will be sequential recruitments of many factors that actually make up the stage through which RNA pol2 binds and starts transcribing. But it will not happen in a reverse way. First, this is not possible that pol2 will come first and then other factors are coming. So this is the importance of directional recruitment. And you can actually use it in any cells. If you have a question and you want to know that two proteins we see they're interacting with each other, and you want to know if which one is a dominant partner, and you can use this assay to verify that.
Wonderful. Also, any of us amongst participant? Okay, no issue. So, Supriya, I hope that you have a good time in Israel doing the research under the pressure of conflict of the neighboring countries. And will stay, and others from other countries gathered in Israel should stay safe and still continue to remain creative and productive. I, if there is, if can we open it to the online? If anybody from other colleagues from any other places from Dr. Ghosh?
Good afternoon, sir.
Yeah, go ahead, sir.
Sir, actually, I want to ask that they started to show you that in that graph, they have taken some hyperparabolic plane. So how they are getting it out that graph they are generating by considering that hyperparabolic means like whatever they are showing that uh, two types of cell type, whatever they have taken. So can I get idea?
Supriya, did you get the question or not?
No, I need to understand which graph you are talking about. The histograms
Yes. Yes.
here. These graphs.
So
It is on.
you
Yeah.
share your screen.
Okay. So can you
Yes. Can
so
you
this
make that zoom or something? It is yeah.
now this
Yeah.
graphs you are talking about?
Yes. Yeah, yes, I'm saying you that like here one hyperabolic like straight line is released. So this one is correlation with anything is there. Yeah, like simply you have
Okay,
considering.
so I will explain you'll understand. So these all pictures are taken in a confocal microscope. Now the confocal microscope that we use here in our faculty is a GIS confocal microscope. Now when we click the pictures, we actually click a G-stack picture, we merge them in a single slice, and then we can there is a software available online that you can download in your computer also, and this is a free software, and you can play with the picture the way you want. Many kind of analysis you can do. So one of those analysis is a co-localization analysis. Okay, so actually this is, I am not doing anything, this is the prediction of the software. Okay, so if I draw a straight line along so this is actually not different filters, this is the same. If you look it very carefully, this is the same cell, and I expose it to different laser, and it gives different fluorescence. So when I give a blue laser here, means for around 300, 350, it gives you DAPI. When I give around 488, it gives HDAC1, means the green signal. When I put it 594, it gives the red signal. And software by itself create a merge picture of this 3 channel on the same cell. So now if I take this merge picture and I draw a line along it, it will automatically in the software, it will draw a line in all these 3 cells, and it will show you the intensity of all the channels, 3 channels you see here in the figure. So you can see here, it's a DAPI channel, and you can see here the intensity of DAPI is low. And you can see from the picture also the intensity of DAPI is low. And it shows the intensity, the green line shows the intensity of HDAC1, and the red line shows the intensity of MECP2. But what we are trying to show here is that you can see the intensity of green and red are merging together. Here you can see the peak are going up together. Then again it goes down. Then again the peaks are going up together. That actually indicates that these two protein, the red and green, are together, but not the DAPI, because you can see the DAPI is straight. So this way, actually software, instead of saying by eye, actually I ask the software to say if they are colocalized or not. That way the reviewer actually will believe, not by saying. If you say that there is a colocalization, no one will believe. You need to show it by software. That's what we did here. I hope it's clear.
Yeah.
Thank you, sir.
Dr. Bhure has got something to say. Dr. Bhure?
Yeah, I don't have anything to ask specifically, but I would like to thank Dr. Supriyo, alumnus of again IVRI. Very nice presentation, Supriya.
Thank you, sir. Very nice to see you. Thank you, sir.
Thank you.
Okay, so if we do not have any other questions from anyone, okay, Shweta has got a question for you, Supriya.
Yes.
Sir, you have talked about that tagness and tag. What, like, it consists of epitopes, you said, no?
Yes.
So what exactly those epitopes are? Like, if we have to change the protein, different protein, then we have to change those epitope also for different
No.
proteins.
If
Common.
no, this is okay. Maybe I will share the screen again.
Yes, sir.
Here. Okay, so this is actually antigen antibody interaction, you can say in some way, but it's not. This region, the small part, the small black below Lana, is actually complementary to those black dots here. Okay, so this is already in a plasmid. What you just need to do, you need to change your protein, whatever you want. In our case, I put Lana. In your case, you can put protein X or protein Y, and then you can easily create dots of any protein in the cell.
Okay, sir.
So,
Is it
so
clear?
Contrary to these questions, is this plasmid free? Is
Yes,
it available?
It's free and available from AdGene.
Okay, that's good. That's good if anybody wants to use it as a resource.
Yeah, if they want, they can contact me directly. I will give you the catalog numbers of those plasmids. You can directly buy from AdGene. From AdGene, it's free. You just need to give the delivery cost.
Okay. With this, I think with this we will close the session here. I profusely thank Supriya for providing us with the latest things, what is going on in cell biology as well as in the cancer biology. Again, thank you so much, Supriya, and we really give you a.
Thank you. Thank you, everyone, for listening to me for 1 hour. Thank you.
Okay. We'll close it.